Hey everybody, in the last video we looked at the telomerase gene, and we looked specifically at a study that was looking at the effects of the telomerase enzyme. I think we would call it a holoenzyme because it's got two parts, right? It has the TERT, It has the TERT enzyme in the TERT RNA. So the telomerase holoenzyme being an enzyme that has multiple components. So now they looked at the effect of telomerase on senescence in cell lines and found out they could take cells that normally senesced after 50 generations and immortalize them by turning on telomerase, by giving the cells a functional telomerase holoenzyme. Essentially, they gave the cells a functional copy of TERT, a TERT gene that was always on. Now, in this video, we're going to take a look at a different study. I believe it is this one right here, Vulliamy et al. Disease anticipation. We're going to call this genetic anticipation. And there's the reference right there. So now what they're looking at is a disease called Dyskeratosis. I didn't think it was called this. Oh, Dyskeratosis congenita. Dyskeratosis congenita. Let me fix that. Yeah, okay, that's right. Dyskeratosis congenita. Now, this is a human disorder. Essentially, fingernails and toenails degrade, fall apart. Skin develops this kind of purplish pigmentation. And there's lots of other symptoms associated with this disorder. The worst one is the bone marrow fails, and this can be lethal right here. I'm guessing treatment would be bone marrow transplant, but I haven't looked up too much more on this disease other than just enough to see some of the symptoms here. Now, in this study right here, they are looking at families that suffer from this disorder, and what we have in these families are heterozygous individuals who are TERT plus, TERT minus. So in the previous video, we looked at the effect of the TERT gene right here. In this one, we're looking at heterozygous humans who are heterozygous for a functional copy of TERT and a non-functional copy of that same gene. So TERT plus and TERT minus. Again, this is the RNA component. Now, okay, so normal humans, wild-type humans, would be TERT plus, TERT plus. Now, these individuals are the ones that develop this disorder. So dyskeratosis congenita. And we are going to abbreviate that as DC. However, not everyone who has this genotype develops this disorder. And that's where I can introduce this term that we haven't introduced at this point in the course yet. It's an important genetic term, and it's called penetrance. So penetrance, we can define that as, let's say, percent of individuals with a specific genotype that also have associated phenotype. Now, I don't know if that's a perfect definition. I like it, but it also helps to see it used. So with respect to DC, Dyskeratosis congenita, let's say, and I don't know if this is true, but I do know DC is incompletely penetrant. But let's say if 50% of humans who have the, let's say, Turk plus Turk minus genotype also show symptoms of the disorder, Dyskeratosis congenita, then we can say the Turk plus Turk minus genotype is 50% penetrant. So essentially, penetrance is a measure of how many of the individuals with this genotype will show the I guess the trait of interest or the phenotype of interest. Not to say that this isn't interesting, but we'll have the disorder of that is the subject of a certain study, a genetic study or genetic analysis. So if 100% of them had it, well, then we say it's 100% penetrant. In our previous examples, we were looking at dominance and recessiveness and sex linkage, and it was always 100% penetrant. This is our first example in this course of something that's not 100%. Now, and so if you look through the notes, you can see there are some other information that, they look at, and you can probably predict that, this is a component of telomerase. And you might predict, well, okay, so maybe these symptoms are caused by, shrinking telomeres. And it is the case. So there is evidence that suggests when they look at people who have symptoms of the disease in healthy individuals, and they measure the telomeres, so telomeres are shorter in individuals who have not only this genotype, but also show symptoms of the disease. And so why can that be? What's going on here? So, these individuals have one functional copy, right? So we've seen this before, right? Where one functional copy is not enough to show the phenotype of two functional copies. So this is a case of incomplete dominance. And the disease probably results from haploid insufficiency. So if this is a chart here, this is levels of the Turk RNA. Let's say this is high, this is low, and this right here is the threshold needed for normal telomere lengthening. So normal telomerase activity. So wild type individuals might have something like this. Maybe there's some variation in something like that. With this would be the standard deviation or something like that. So wild type individuals, Turk plus, Turk plus. Now, Turk plus, Turk minus heterozygous individuals, this copy could be null. Makes a totally non-functional RNA molecule. Maybe the RNA molecule is mutated, so it's non-functional. So they may only express this much of the functional Turk plus RNA. So telomerase activity is not normal, and as a result, the telomeres will shrink over time. Now, we also saw something else interesting from this study. So this is a pedigree from the study looking at 1, 2, 3, 4, 5, 6, 7, 8 different families. So black shaded individuals, males and females, these show symptoms. They have the disorder, symptoms of DC. And they also have the Turk plus, Turk minus heterozygous genotype. Now, unshaded shapes are wild-type genotypes. Gray shapes, so these are the Turk plus Turk minus genotypes that do not show symptoms, do not have symptoms. They may get it in the future, the disorder, but at this point in their lives, they do not show symptoms. So if you look at all of the black shapes here, there are numbers. And those numbers represent the age that the individual started showing symptoms of DC. Now, the gray symbols here, they have ages in parentheses. So those are the ages of these individuals at the time of the study. And what we talked about in class, and I think was one of the main points of this paper, is that with respect to this disease, this disorder, DC, that it looks like the children of parents who are heterozygous for disorder are showing the disease at much younger, symptoms of the disease, or getting the disease at much younger ages than their parents are. So look at, this is a good example right here. So this individual is heterozygous and is technically susceptible to the disease, but hasn't developed it yet and is 61 years old. But he has a 5-year-old or 9-year-old son who's already developed the disorder. Again, over here, 60-year-old right here, so, or has a son that developed it at 9 years old. This individual here, daughter and a son, both developed the disease at 20 years old. This individual here, 48 and 46, so both of them, or these individuals, both of them had children that developed the disorder at 13 and 16. And well, 37 over here already has the disorder, developed it at 37 years old. This one has a child that developed the disorder at 3 years old. So, and the idea, well, what we think we're seeing is genetic anticipation. And genetic anticipation is a term for a disorder that appears earlier in the offspring of parents who have the disorder or are susceptible to the disorder because of their genotype. So the disorder is appearing earlier and earlier each generation with genetic anticipation. And we'll see another example of this when we look at trinucleotide repeat disorders in a future lecture. So that's genetic anticipation, and it could be what's going on here is that the germline cells in these individuals lack adequate amounts of functional telomerase holoenzyme. So they're passing, in this case, this individual right here is passing sperm with chromosomes who have very short telomeres. So in each of these cases, the children are starting with chromosomes that have telomeres that are very short. So all of their cells in the bodies of these children as they develop are starting with telomeres that are just really short, much shorter than what their parents had to start with because of their parents lacking the necessary enzyme to lengthen the telomeres. And that's one hypothesis to explain why genetic anticipation seems to exist with this disorder, Dyskeratosis congenita. Okay, so I'm on my way to a meeting. I got 2 minutes to get there. I hope you enjoyed this segment of our course on telomerase. It's as I think an interesting aspect to talking when we're talking about DNA replication in eukaryotes, telomeres and increasing human lifespan, and I think it's all really interesting stuff. I hope you do too. Again, take a look at the notes. All the stuff is laid out much more clearly and in a much more organized way in the notes, and try to answer the problems that are present in the lecture notes to help you study for the exam. So see you at class on Thursday or office hours, or email me if you have any questions. Okay, see you later.